Hi everyone, a very warm welcome to the FARRE series of 2026. Those of you who have joined, please come and say a hi in the chat box. I'm eagerly waiting to meet all of you. Please come and say a hi. Just yeah. Okay, all right. Hi guys, good evening, good evening. Hi, NEET aspirant. Okay, Gurpreet, I think you should go and read your physics, chemistry and biology, and very soon I will see you in on the other side in these MBBS second year lectures. This lecture is meant for the MBBS second year university exams. So hi guys, good evening. Good evening Siddhi, very nice to see you as always. Chaliye, so we are beginning with the FARRE series. Not beginning, this is the second lecture. Today I am taking immunology. Good evening, Nishu. Good evening, everybody. All right. So, immunology करेंगे आज in FARRE we are going to revise these particular topics. We are going to actually revise in the form of questions which are commonly asked, है ना? So major topics, the basic orientation and discussion. Why have I pasted this one shot immunology which I have done on the Medit page itself? In this we have covered many important aspects and topics. So many of you know. Hello, hi guys. Good evening. Very good evening. So, सबसे important topic antigen, types of antigen, adjuvants, T cell dependent, independent antigen, haptens and super antigens. All of that cover in the one shot. Immunoglobulins or antibodies, types of antibodies, the GAMDE, 5 types of antibodies, basic structure and diagram of an antibody. Then what are the functions of the different 5 classes or isotypes of antibodies. All of that also discussed in this lecture on the one shot in immunology. Then also the antigen antibody reactions are also covered in this lecture, which is the precipitation reaction, agglutination reaction, complement fixation, neutralization, and the newer techniques such as ELISA and lateral flow assay. It's a wholesome lecture over there. So I hope, as many of you have watched it there as well, I hope that you have covered all those basics from this particular lecture. Today in this FARRE class, we are going to discuss primary versus secondary immune response, because this is exactly how they ask you to write the differences table in the exam. Okay? I hope by this time most of the second year students, they know, they understand what is primary immune response, what is secondary immune response. But when they are asked to make a differences table, it is very difficult to come up with those 5-6 points of differences, right? Micro is really interesting. All right. Thank you so much. I agree with you as well. I also feel that microbiology is very interesting, and that is my purpose as well. So, yes, and the second is active versus passive immunity. वो थोड़ा shift करना था. Active versus passive immunity. You also know active का मतलब क्या है, जिसमें the immune system has to do some hard work, whereas passive immunity is when the immunity does not have to do any hard work. Passively we are injecting something from the outside, but again the differences table becomes a problem. This is like the most controversial complement system, because mostly it gets ignored, because in microbiology the teachers say, oh, you have read it in biochemistry. In biochemistry the teachers say, oh, you read it in microbiology. So nobody ends up teaching, nobody ends up learning. Also it's there in physiology, pathology. This one particular topic is there in 4 subjects of first and second year MBBS, but it always gets ignored. So complement system we are going to do in detail, we are going to understand, and इतना simple कर लेंगे, इतना easy कर लेंगे कि you will wish and pray कि exam में complement system पूछ ले, so that we can do it. CMI versus humoral immune response. Again, like I said, you know कि भाई cell mediated का मतलब T cell है और humoral का मतलब B cell है, but exam में कैसे लिखें, how to make that differences table. MHC, ये अगेन अनदर वेरी इंपॉर्टेंट कॉन्ट्रोवर्शियल टॉपिक. आज कभी किसी को क्लियर ही नहीं हो पाता what is major histocompatibility complex, what we are trying to say, and then usually between the 2 classes of MHC you are asked to differentiate. Mostly we just end up thinking that MHC is the receptor present on antigen presenting cell. अब इसके आगे what to write in the exam, right? So that is why we are going to do that. Then hypersensitivity reactions as well. all the 4 types of hypersensitivity reactions and the beautiful mnemonics to remember all of these things. Okay? So we are going to cover all of these topics in this particular FARRE session. Like I said, all the different types of antigens, super antigens, adjuvants, T-cell dependent, independent, antibodies, classes of antibodies, precipitation, agglutination, complement fixation, neutralization, ELISA, everything has been covered in this lecture on the one-shot series of immunology. Alright, so you can go and watch that as well. So like I said, primary versus secondary immune response and humoral versus cell-mediated immunity. Also also in that same lecture on the one shot, the innate versus acquired immunity has also been discussed. Just to give you a wholesome idea, just to begin the lecture, that alright Nishu, it's good to know that you know that MHC matching is required before organ transplant, but we are going to discuss the details of it, so stay tuned. Right. So you know that when I talk about immunity, there is the acquired aspect and the innate aspect. The innate immunity is something that we are born with. Innate immunity contains of the cellular components, the chemical components, as well as the external barriers. External barriers includes, of course, the skin, the mucous membrane, the saliva, tears, gastric juices, etc. The cellular components involves the most important natural killer cells and the phagocytes. Phagocytes ka examples include macrophages and your neutrophils. What do? They come and they eat up the pathogen which has entered our body. Then of course you have chemicals such as interleukins and cytokines and acute phase reactants, and then you have some physiological changes in the body. Why do you get a fever? Why does the temperature suddenly rise? It is all of it is the innate capacity of my body to kill the pathogen. Right? Now all of this is innate. This is what I am born with. This innate immune response does not डिफरेंशिएट बिटवीन द टाइप ऑफ पैथोजन अटैकिंग. ठीक है, इसको नहीं फर्क पड़ता कि ये वायरस है, बैक्टीरिया है, फंगस है, क्या ये कोई वो पैथोजन है जिस व्हाट वी हैव सीन बिफोर हैंड, और इज इट अ न्यू पैथोजन विच हैज एंटर्ड द बॉडी फॉर द फर्स्ट टाइम, और इज इट समथिंग विच हैज एंटर्ड बिफोर हैंड एंड नाउ अगेन इट इज अटैकिंग. innate immunity does not care about all of this, right? Innate immunity doesn't care. So what does innate immunity do? It mounts the exact same type of immune response all the time, every time any pathogen comes, irrespective of how many times that pathogen has entered inside the body. Then you have the adaptive immune response, and I always give the famous Dhurandhar line for adaptive immune response, घायल हूँ इसलिए घातक हूँ. So why is adaptive immune response a much larger or a heightened immune response? Why is it a better immune response? It is a calculated immune response which is very, very sophisticated, and it only happens in case there has been a previous exposure to the antigen. ठीक है, घायल हूँ इसलिए घातक हूँ, meaning I have been hurt before, that is why I am dangerous today. So that is what adaptive immunity is. It has already seen the pathogen in the past. Guess correct, Siddhi. It has already seen the pathogen in the past, and now they have recognized the pathogen. It has created a memory, and now it is mounting a very specific and a very intelligent kind of an immune response onto the pathogen. So that is the understanding of innate versus acquired immunity, right? If you're supposed to write the differences, you can very well refer to that lecture. Now then, the second thing that they ask you is primary versus secondary immune response. You understand primary immune response is something which is happening for the first time ever. So that means whenever a pathogen enters my body for the first time, okay, I was having a fun time in my life, went to Pani Puri vendor, ate nice Golgappas and Pani Puris. Vendor was a carrier of E. coli, gave me lots and lots of E. coli. For the first time ever, this particular strain of E. coli has entered my body. So what will happen immediately? The thing about primary immune responses or pathogen entering for the first time in the body is that the body is not prepared. Body has not seen it beforehand. So there cannot be any antibodies around floating about. You know, humans have not made antibodies against all possible pathogens in the world. The only way that humans make antibodies is once they have seen the pathogen, it has been presented to the adaptive immunity. Adaptive immunity has started making the antibodies, then we get it. So what happens in primary? It is against the first antigen encounter. So the first time when the antigen or the pathogen enters my body, the first response is the primary immune response, whereas secondary is against a subsequent antigenic exposure. Now, it is very important to write subsequent. Why am I highlighting the word subsequent? Because secondary immune response only comes when the same pathogen attacks again. Guys, it's not that some other pathogen. So, please don't write when the second time or another time, don't use these words. Please use the word subsequent antigen exposure. Why am I focusing or emphasizing on the word subsequent antigen exposure? Because the same antigen or the same pathogen has attacked again. Okay, same pathogen has attacked again. That is when my body will give a secondary immune response. And like I always give you the example of a one tight slap. So the first time you get a tight slap, you had a lag phase. You know, you're like, okay, what happened to me? What happened? Then you take some time and then you realize, and then you have anger building up. Now you're prepared. Second time also, if the tight slap comes, oh, you're going to really hit hard the other person. That is what secondary immune response is. Subsequent attack, same antigen attacks again. Now the body loses it. Now the body gives a very heightened secondary immune response. No, Nishu, it is not called as that. Please understand. Amnestic response is totally different. You should not give out words like that. Okay, secret masala, yes. So, okay, amnestic response is a phenomenon which is seen in the Vidal test in the laboratory when the antibodies are made against some viral pathogen, but they end up cross-reacting with a bacteria. Okay, so that is a totally different story. Please don't mix the two. Chaliye. So we are talking about primary immune response. Like I said, the first time you get a slap, you are confused. There is a lag phase. So that is why, jaise hi first slap comes, you take some time to become angry. That is slow response. Similarly, the first primary immune response happens. It is slow to appear and it is short-lived, right? It happens slowly, and then the body forgets about it, that okay, probably it happened for the first time. Here the lag period is longer. Lag period is longer, right? So we can always draw it like this, that whenever there is a primary attack, so you should always make this graph in your answer sheets, and you should say, यहाँ पे हुआ first attack or primary attack. So what happened? Immediately you went into a lag. You did not realize what happened. So there is a lag, glitch ho gaye, you are glitching, you don't know what's happened, you're confused. Suddenly you start feeling angry. So slowly your anger increases and it reaches its peak, right? Then you are angry for some time and then you forget about it eventually. What is this? This is the primary immune response. What is this whole thing? This is the primary immune response which is happening in response to the pathogen for the first time. So I am saying that the lag phase, what is the lag phase? This is the lag phase. It is there, and then the first antibody which rises, if at all there is an antibody response, it is mostly always IgM. So it rises and it falls after some point of time. ठीक है? चलो. Lots of love to you as well, Ali's. Very nice to see you. All right, chalo. So I said it is slow to appear and it is short-lived. The lag period is longer and there is no negative phase. What is this negative phase that we are talking about, and that is how we are differentiating? Slow to appear, short-lived. Here it is prompt, powerful and prolonged response. Here the lag period is absent and a negative phase may occur. What is the meaning of all of this? That is what I am trying to show you with that graph. What is the meaning of all of this? So क्या हो रहा है? First attack आया, there is a lag, you are glitching, you don't know what has happened. Then there is an anger rising, the antibodies rise, they stabilize and then they fall. Now, they were falling nicely, okay. They were like, okay, fine, I will forget this person. Now, this antigen is a idiot. Antigen comes again. Second attack by the same antigen, right? So, this is subsequent attack. Now, the body is going to lose it. It is going to have a very prompt and very high response. So, what you are expecting is, the moment at this time this happens, you are expecting that the body will shoot up and give a very, very high immune response. Yes, this happens. A very prompt and shooting up and a high response happens. But what happens before that is, see, again the body was settling down a little. है ना? Your gussa was going down a little bit. You were just trying to relax. But suddenly it happened again. So now, whatever residual anger you have, first response is going to be with that, right? So the first slap is always going to be lighter and then the subsequent slaps are going to be very, very strong. So that is what happens. At this point of time, these antibodies which were circulating in my blood, they had not come down to a zero. They had fallen for sure, but they were still above zero. these circulating antibodies were floating about in my blood just in case a second attack happens. And that second attack happened. So whatever the circulating antibodies were, first they are going to get consumed and dip, right? And then suddenly the second immune response is going to come. So what is this happening here? It is this dip that I am talking about. Why does this dip or the negative phase happen? Because the circulating antibodies are used up in the very first time. The very first time all the circulating antibodies get used up. There is a slight dip and suddenly your adaptive immunity gets activated and you get a heightened, very prolonged and a very strong immune response. So this is the negative phase or the dip that I am talking about. So when you compare, so this is, what is this? On the other hand, this is the secondary immune response, right? And mostly these antibodies are IgG type. So like I had said, there is, how big is the lag phase here? Quite big. Is there a lag phase? Almost nil. Is there a negative dip anywhere? No. There is a negative dip, negative phase? Yes. Then what is the extent of the immune response? The immune response is slow to rise. See, this is rising at a slant, slow to rise, whereas this one is sharp rise. Yes. So it is slow to rise, and it is short-lived. It falls down also. This is sharp to rise and it stays risen for a prolonged period of time. So now can I go back and see that all the words that I said were correct or not? Yes, they were correct. It said it is slow to appear. We saw the slant. It said it is prompt and prolonged. Yes, it is prompt and prolonged, immediately happening and staying risen for a long amount of time. Then lag period is longer, lag period is absent. There is no negative phase in primary immune response, correct. There is a negative phase in the secondary immune response, correct. So these are the differences that you will write. Good evening, right? Yes, IgM always appears first. Now, antibody low titer and is of IgM type. Yes, if you compare the absolute titer of antibodies, you will realize that this graph is much lower as compared to this graph. Yes, so the total antibody levels, titer means total antibody levels, is far less in the primary immune response as compared to the secondary immune response, right? So the antibody titer is low and usually of the IgM type, because IgM is the first antibody to rise, and here antibody produced is in high titer and is of the IgG type. As you can see, it was heightened and it was IgG type. Antibody produced here in the primary immune response are less specific because they don't have much time. So whatever floating IgM antibodies are there, they are going to come and attack first. But in the secondary immune response, you have a more specific type of attack because memory has come into play. You've gotten the time, the body is prepared from beforehand. Here antibody producing cells are the naive B cells. Here the antibody producing cells are the memory B cells. Like I said, memory B cells are always going to have a more specific type of an immune response. So here it's a naive B cell. It's a naive little kid. They don't know that they can get slabbed also, right? So they were not sure that this can happen, but it happened. Now they have become memory. Now they know that this can happen and immediately they are going to mount an immune response. यहाँ पे क्या हो रहा है? It is both T dependent and T independent antigen. Over here, only T-dependent antigens are processed. I hope you all know what is the meaning of T-dependent and T-independent antigens. It has been covered in this lecture. I have covered T-dependent and independent lectures, antigens at length. If you want me to explain, please write in the chat box. I can quickly explain the T-dependent and independent antigens. But I hope you know that a T-dependent antigen is the one which is the protein antigen. Whenever a T cell dependent antibody response comes, then a memory is formed. Whenever a T independent response comes, a memory is not formed. T cell independent response basically means the B cell is identifying the pathogen, the B cell is producing antibody, and that's it. A T cell dependent response means a B cell is seeing the antigen, but it is not able to process it. Then the B cell becomes an antigen presenting cell and presents it to a T helper cell. Now T helper cell will again activate the B cell and then the antibodies will be produced. So this catching the ear like this is the process which is called as T cell dependent antigen. And always a better immune response, a specific immune response, a prolonged immune response and memory formation happens whenever this long process of T cell dependency takes place. I hope it is clear. If you want, I can draw and let you know. Otherwise, we shall continue because you can always watch it in that lecture. So, number 1, immune response. When does it happen? 2, slow lag period negative phase antibody titers. Then you will talk about specificity of antibodies, nature of the B cell and nature of the antigen which is involved. So, antigen involved is T cell dependent or T cell independent. Alright. So, this is a diagram that you will make for primary and secondary immune response. Another beautiful diagram that you can make which will actually tell you how the IgM and IgG they behave in primary and secondary immune response is this one. So, I am saying that this is primary immune response and this is incorrect, IgM. Yeah. So, the first exposure to an antigen happens, immediately the IgM rises and falls. Whenever there is a second exposure on the second immune response at a later date which happens, here the IgM rises later than IgG. So, always in second immune response, the IgG is always higher or it always comes before IgM. If you are more confused, let me tell you. So, what will happen whenever it is a primary immune response or the first attack in your body? You know this. Whenever there is primary attack or first attack, the first antibody which comes is IgM, then it settles after some point of time, right? And now, after some time, a few weeks, your IgG rises and it stays high forever. This is what you know normally, है ना? सबसे पहले IgM rises and falls and then IgM is replaced by IgG and that is what forms your memory. What happens in a second immune response? Suppose again you got attacked by the same pathogen. Now this IgG was already floating about. IgG is prepared. You have memory. So before IgM, your IgG is what is going to rise. Yes. So even before IgM has a time to rise, this IgG rises very, very fast. And then after some time you have a rise and fall in the IgM like normal. Okay? So these diagrams, graphs can also be made and I would want you to make or draw this particular graph or diagram because it is very, very, suggestive and a very good diagram to draw in the exam. Chalo. Next pe aate hain. What was the next topic? It was active versus passive immunity. Yes, active versus passive. I have always told you what is active immunity. It is the immunity in which my body has to work hard. So when does my body has to work hard? When it has to see the antigen, process the antigen, take it to a helper cell, show it to a helper cell. That helper cell has to release cytokines. Those cytokines have to go and activate B cell. B cell has to activate plasma cell. Plasma cell has to produce antibodies. This is an active process, right? What is passive process? Body wasn't doing anything. You took an injection, you filled it with preformed antibodies and you injected inside a person. Now these antibodies which have been created artificially or naturally but not by my body have been injected from outside inside my body and therefore my body is able to attack the pathogen. This is passive immune response. Now both of these active as well as passive immunity can be natural or artificial. Both of them can be natural or artificial. For example, active immunity is produced actively by the host immune system, whereas passive immunity is immunoglobulins. Immunoglobulins means antibodies itself are received passively by the host. By me, I am the host. Okay. Now, here, how can active immunity be induced? By infection, which is a natural source, and by vaccination, which is an artificial source. Yes, so what happened? I am living my life naturally, a bacteria or virus enters my body. Now, the whole thing starts. My B cell has to see it, show it to T helper produce cytokine release, B cell, plasma cell, antibody, all of this happens. Or, a doctor took a little piece of that microorganism and injected in the form of a vaccine. You know what are vaccines? Vaccines can be a live but attenuated organism. Vaccines can be a killed but a whole cell of an organism. Vaccines can be a little subunit taken from that organism. Vaccines can be the mRNA taken from that organism. Vaccines can be, so all of these are the models of vaccines, the live vaccines, inactivated vaccines, injectable vaccines, killed vaccines. Yes. What are these vaccines essentially? You are taking a little part of any antigen, or you are taking a whole organism and you are injecting inside. Now my body doesn't know that it is a infection has happened through natural sources or artificial sources. Body has to still process that antigen, activate B cell, T cell, etc. So now that is active. So it can happen naturally in the form of infection which I take from nature, or in the form of vaccine, which is an artificial form of infection under controlled environments. It is definitely long lasting. Why? Because when my body has undergone all of this processing in the same path, what has it created? It has created the memory B cells also. Because when do the memory B cells get made? Like I said, whenever an antigen comes, the B cell takes it, shows it to T cell produces cytokine, cytokine activates B cell. In this process, the memory cells get created. And when the memory cells will get created, the immune response is going to be long lasting. That is why in your active immunity, there is a long lasting immunity. Yes, of course, there is a lag period which is present in active immunity. Like I said, you get slapped for the first time, you'd glitch for a bit. You take some time to get activated. So all of this process is taking time. So there is lag. Of course, memory is present here, like I said, and it is not useful in immunodeficient individuals. Why? Because the immunodeficient individuals are immunodeficient because of a problem in any one step, they are not able to take this whole process. Yes, either they don't have B cells, or they don't have T cells, or there is some cytokine, or there is some receptor deficiency. So all in all, this whole process they are not able to do. That is why active immunity is produced actively by host immune system, induced by infection or vaccination. It is long lasting, it has a lag period, there is memory, and it is not useful in immunodeficient individuals. Whereas passive immunity is acquired from outside. Body doesn't have to do any hard work. So no B cell gets activated, no T cell gets activated, no cytokines get released, nothing happens. Passively, the army from outside has come, killed the pathogen and left. Your own police, your own army is inactive, right? So here it is acquired by mother to fetus, where IgG is transferred. So my students would know, we have the beautiful mnemonic, Mummy G. So Mummy G, Mummy G. So mother gives IgG antibodies to the fetus. So passively they have come. Fetus's body has not worked hard. Passively, because of placenta, the antibodies have come. Or ready-made antibody transfer in artificial immunity, such as immunoglobulin injections. जैसे कि anti-tetanus serum, anti-venom serum, your immunoglobulins against hepatitis B. What is all that? Outside in the laboratory, inside an injection, I have preformed antibodies which I will inject. Right? So natural sources is obviously भगवान का दिया हुआ, and artificial sources is जो भगवान नहीं देता वो डॉक्टर देता है. Right? So all of that is doctor induced. Now passive immunity lasts for a long time, for a short time. क्योंकि हमने कोई मेहनत नहीं करी है, कोई मेमोरी नहीं बन रही है. बाहर से from outside some antibodies had come, done their job. Antibodies are not, immortal. Antibodies are also proteins. Eventually, they also get degraded. But when my body has done the mehnat, again and again it can keep doing the same thing. Right? But what has passive immunity done? Antibodies came from outside, did their job, got degraded, vanished from the body. Right? There is no lag period. Lag period hai kya? From outside they have come, they were prepared to attack. So there is no lag period because there is no antigen processing involved. Processing is not involved in this. Of course, no memory will be formed because all of that shebang of B cell, T cell and everything is not happening. And it is useful in immunodeficient individuals, right? So immunodeficient individuals who don't have the ability to do all of this process, in them it is very, very useful because we just send antibodies from outside and they act like treatment and they cure the patient. Active natural exposure to antigen, active passive vaccination, passive natural mother to baby, IgG, IgA, passive artificial ready-made antisera, करेक्ट, रेबीज की स्पेलिंग गलत हो गई बस, दैट्स ऑल इज राइट. IgA भी तो मदर से बेबी को मिलता है. यस मिलता है. थ्रू द ब्रेस्ट मिल्क इट इज ट्रांसफर्ड. चलो, okay, all right. So like I said, active versus passive, this can be asked to you and you have to write it in these particular points. 1 2 3 4 5 6. 6 points you will write in that. Now we have come to the most important topic of today, which is the complement system, and let's quickly revise the topic. So are you ready? Shall we begin? So when I say what is complement, complement is a series of enzyme activation which leads to ultimately a membrane attack complex formation or opsonization and killing of the pathogens. ठीक है? What is complement system? Complement system is a system which contains of some inactive enzymes which get activated in a cascade, wherein the previous reaction, the next reaction is always larger and over and above and beyond than the enzyme which activated them. So what is happening? There is one guy, okay? There is one guy who is sleeping. Who is he? He is any enzyme. He is any complement enzyme. Good evening, good evening. Right. He is any complement enzyme. Now, something happens, a kick happens. This guy gets kicked. Now suddenly he is woken up. Now he is kicking other guys. The other guy that he kicked, that gave me a bigger reaction. When this guy kicked the other guy, even bigger reaction came. He kicked another guy, even bigger reaction came. So what is this? This is a cascade effect. When one thing leads to another, there is a snowball effect. That is what is complement activation. What happens in complement activation? There is a cascade effect, cascade or a waterfall effect, wherein inactive enzymes get activated and every enzyme which is activating the next step, it is leading to a higher and a larger reaction. And it is like a chain reaction. So all of these complement enzymes, what are these complement enzymes? These are inactive enzymes which need to get activated by some kick. Yes. So by some kick. How many complement enzymes are there? 9. So you have C1, C2, C3, C4, C5, C6, C7, C8 and C9. Okay. These are 9 complement enzymes which are always floating in our blood. They are inactive, but they are always floating about. Yes, it's not that something will happen so they will come. No, they are always floating. They are inactive. Yes. Now whenever they get a kick, suppose C4 got a kick. When C4 gets a kick, it breaks into 2. So whenever one complement gets a kick, it breaks into 2. Now C4 is going to break into 2 portions. So this is C4, it will break into this one big segment and a small segment. Yes, the big segment is always B for big, is the B segment, C4B segment, and the smaller segment is always segment A. Oh sorry, it's C4. Smaller segment is always segment A. Okay, so B is always bigger and B is what we call as the binding unit. The binding unit or the binding segment. What will the binding unit do? It will go and kick the next enzyme in the cascade. So suppose C3 got activated or suppose C4 got activated. So when C4 got activated, it broke into 2 parts, C4A and C4B. C4A is anaphylotoxic. What does C4A do? C4A says, what is is calling others, come, come, come. So what does C4A call? It calls anaphylactic response. It calls the mast cells to degranulate. It calls the histamine, the vasoconstrictors. So A is for anaphylotoxic. So always the A segment is anaphylotoxic. It floats away. It floats away. So remember A and away. What is A? A is anaphylotoxic. Away is floats away. What does B fragment do? B fragment binds to the next guy. So the C4B will go and bind to C5 and break C5 into 2 parts. C5b will go and bind to C6. So that is what keeps happening. One complement enzyme, when activated, breaks into two parts. The A segment is anaphylotoxic and floats away. The B segment goes and binds to another complement in that path and breaks that into further two parts, right? In all of them, the C5b is always the bigger fragment and the binding fragment, except C2, in which the C2 in which the C2a is what is a binding fragment and a bigger fragment, and the C2b is what floats away and is anaphylotoxic. So except C2, all of the complement enzymes follow the rule of C4, which is B is binding and A is anaphylotoxic, right? Shobhit, I will try to revise all the topics of microbiology. What I am trying to do is, if I have covered any topics in one shot series or previous year's FARRE, I am not covering in this year, so that you can watch those lectures and then come and watch this lecture, so that maximum chunk of the syllabus gets completed, right Shobhit? I mean, before asking this, you should have thought, as a second year MBBS student, how is it possible to cover entire microbiology in 2 2-hour segments? But we are really trying to cover most of the things. It is definitely possible to revise entire microbiology in FARRE series. It is not possible to cover and explain entire microbiology in FARRE series, alright? So, I will try my best to revise maximum as much as possible. So, in the beginning of the lecture itself, I had said that the other topics which I am not teaching have already been covered in the one shot lecture, and the rest of the topics I am teaching in this lecture. And as we start with systemic microbiology, where I will teach you bloodstream infection, GIT, etc. In those also I will make sure that the topics which I covered last year, I will do fresher or newer topics this year. सही लिख रहे हो है ना? Complement system, all right, correct rabbit, hai na? So C1 to C9, this is how the cascade works. Now another thing that we have to note is that the complement pathway is of 3 types. Yes, so we said what is the starting point? A starting point can be any trigger or any kick is going to start the cascade. That is the starting point of the complement system. What is the end point of complement system? No matter the starting point, no matter where we have started from, no matter if we started from classical pathway, no matter if we started from alternate pathway, no matter if we started from lectin pathway, ultimately we reach a common point. What is that common point? That is MAC formation, which is membrane attack complex formation, or opsonization. These are the 2 end results of complement pathway. So what are the 2 end results of complement activation or complement cascade? It is MAC formation, membrane attack complex formation, and it is number 2, opsonization. Opsonization, and you know what is opsonization? It is the process by which my body makes the antigen tasty for the phagocytes, to eat up. Yes, and of course, like I had said, the R is always going to go away and lead to anaphylaxis. So what is the third step which happens? It is anaphylaxis. Okay, and complement system also becomes a bridge between your adaptive and immune response. So the fourth thing which happens is that it becomes a bridge between adaptive and innate immune response. So you can write all of these points also, that what happens ultimately as a result of this pathway, it is MAC formation, opsonization, anaphylaxis, and bridge between adaptive and immune response, innate immune response, and ultimately there is killing of the microorganism, right? Killing and destruction of the microorganism. So like I said, we have to know that there are 3 types of complement systems. You have the classical complement pathway, you have the lectin complement pathway, and you have the alternate complement pathway. ठीक है, classical, lectin and alternate complement pathway. Classical का अब सब का what is the major thing, the major difference between the 3? The starting point of all 3 is different. ठीक है, all three have a different starting point. So what is the starting point of classical pathway? That there is something which is C1, right? So what we will do as we are reading the complement pathway, we will make sure that we have covered or used up all the 9 complements. So in the classical pathway we are going to start with C1. Yes, so we are going to consume and utilize C1 as the first. Classical में क्या होता है? C1 gets consumed. What comes into play? C1. So let's suppose that the C1 looks something like this. Okay, it's like a 6-unit bunch of flowers. ये है C1. C1 में क्या होता है attached? It has some proteases and esterases attached to it. ठीक है, तो इसके साथ में ऐसे ये 2 कुछ enzyme type लगे हैं, which are the proteases and esterases which are attached. What are these? These are CR and CS. So what are CR and CS? They are nothing but proteases or esterases. You understand that a protease or an esterase is going to break a protein or an enzyme. Yes. The moment the C1 gets activated, the CR and CS, they rotate. When they rotate, they activate this C1. Right? The moment they rotate, they activate this C1. So let's look at how this is happening and actually how does the classical pathway come into play. I am telling you that out of all of these pathways, classical pathway is associated with a secondary immune response. ये किसके साथ है? ये है आपकी secondary या adaptive immunity का part. It is a part of adaptive immune response. Whereas lectin and alternate both are a part of the innate immune response. ठीक है, सो क्लासिकल का नाम क्लासिकल है, इट इज कॉल्ड क्लासिकल, बट इट इज इन फैक्ट एसोसिएटेड विद अडैप्टिव इम्यून रिस्पॉन्स एंड इनेट lectin and alternate are associated with the innate immune response. So let's look at the classical pathway. What is going to happen in classical? Like I said, it is associated with adaptive immune response. So don't you think that an antibody is going to play a major role? So let's imagine that this is a pathogen, this is a microbe which has entered the body. The microbe on its surface has this antigen. कुछ भी है, it's some bacteria, virus, anything. It has this red colored antigen on its surface. The second time this has happened, right? This is an adaptive immune response. So who is going to come into the picture? What is going to come and bind to this? My IgG antibody. So who is the guy who is going to come and bind? It is the IgG antibody. Who has come? IgG. The moment IgG binds, my C1 gets activated. So what is the starting point of classical complement pathway? What is the first thing that happens? It is antigen antibody complex formation. Antigen antibody complex formation is the starting point of classical pathway. That is why it can only happen in adaptive immune response. ठीक है ना? Antigen antibody complement complex is the starting point, is the trigger for classical complement pathway activation. ठीक है? इससे आके क्या बाइंड किया हमारा? इससे आके हमारा बाइंड किया C1. C1 with its CR and CS has come and bound to our, ये आ गया C1 and it is bound to the antigen antibody complex. Now this is going to get activated. How do we denote an activated C1? We activate it with a bar on top. So C1 along with its CR and CS unit have gotten activated. So this is an active C1. What is this? This is an active C1. Alright. जब ये C1 एक्टिव हो गया या एक्टिवेट हो गया, that is going to kick start another pathway. अब ये आगे किसी को किक करेगा. Okay, it is going to kick something. So I know for now that what is the starting point of classical complement pathway? It is the antigen antibody complex formation. Is the starting point. What happens in lectin and alternate pathway? In the lectin pathway, the starting point is the mannose binding lectin. What is mannose binding lectin? So in my body, there is another such bouquet of flowers. ठीक है? What is it? It is a lectin which binds to the mannose present on the surface of bacteria. What is mannose? Mannose is a sugar. It is present on the surface of bacteria. What is this guy? This guy is a lectin which binds to the mannose. So can I call it as mannose binding lectin? What is the short form of it? MBL. MBL is the mannose binding lectin. Okay. What does this mannose binding lectin or MBL have on its surface? It has two more proteins. So यहाँ पे इसके साथ भी attached है एक ये pink color का बंदा और एक ये green color का बंदा. These pink and green molecules are attached to it. What are these? These are mannose activating serine proteases. Have to be proteases, yes. So short form of that is, what is this? This is mannose activating serine proteases, MASP. So everything needs an activator, yes. So here C1 ka activator was antigen antibody binding, then CRCS activating and activating the C1. Over here, the mannose binding lectin binds to the mannose and the MSAP comes into picture, and it is also a type of a protease, so that also activates, right? So what is the activation point here? What is the activation point here? Here it is happening because of mannose binding. Mannose binding is the starting point of the lectin pathway. Yes, so we can also draw this in fact. So what was happening in the lectin pathway or the mannose binding pathway? Here is this bacteria. This bacteria has these mannose residues on its surface. What are these orange proteins? These are the mannose. Mannose. What is going to come and bind here? My mannose binding lectin. Yes. Who is going to come and bind? The mannose binding lectin is going to bind to this mannose, right? Then the MASP gets activated. So when the MBL and MASP get activated, what are they going to produce? An activated MBL. So I will write that with a bar on top. This. Now this is an activated MBL and MASP. This will go and further activate something which we will see in just a minute. Yes, I hope it is getting cleared up till now. There is no confusion up till now. It is quite simple and quite clear. Now let's look at alternate pathway. What happens in alternate pathway? It is completely alternate. It is completely different. What is happening here? Here, the C3 was there. Who is C3? C3 is this guy. C3 is this guy. Now you are saying, how come suddenly C3 is getting activated? Like I said, all of them are floating in the blood only, even though inactive. Yes. So when the pathogen entered my body, C3 was also there. C3 was floating about inactive. Suddenly C3 realized that there is some virus, bacteria, fungus, some pathogen is there. And the moment C3 sees any pathogen, it undergoes spontaneous hydrolysis. Spontaneous hydrolysis. Apne aap, on its own. Nobody said anything to C3. The moment C3 saw bacteria, fungus, virus, lost its mind, exploded, hydrolyzed on its own. When it hydrolyzes, it breaks into, of course, C3A and C3B, right? Now this C3A and C3B is going to further get activated and lead to something. Okay? So like I said, what can happen in classical, yes. So quickly let's draw alternate pathway. What is happening in alternate pathway? There was anybody. Either there was this bacteria with its lipopolysaccharide, or there was this fungus with its proteins, or there was this virus with its own surface proteins. So the moment C3 saw any of these guys, this bacteria, this fungus or this virus, C3 which was floating about normally lost its mind, spontaneously hydrolyzed into two fragments. Yes, into a bigger C3b fragment and a smaller C3a fragment. Correct? So what is going to happen now? You know that B is a binding unit. Yes, and A is an anaphylotoxic unit. Now this C3A is going to go away, cause anaphylaxis, that is all right. है ना? यहाँ पे क्या हुआ है? यहाँ पे हुआ है spontaneous hydrolysis, and the C3A has gone to cause anaphylaxis. What will C3B do? It has to bind to something. It is waiting to bind to something. What happens in the alternate pathway, which is a part of innate immune response? The moment this reaction happens, there is another factor floating about, which is called as the B factor or the factor B. Okay? This is also floating about somewhere in the body. The moment this factor B sees that C3 has broken into two components, this B also breaks into a big and a small factor. So the big is the BB and the small is the BA, right? You know what is BB going to do? BB is going to bind with 3B. So ultimately what will happen? These both will go and bind to this pathogen. So suppose, let's take the example of this bacteria. This bacteria was sitting there with its lipopolysaccharide, which had spontaneously hydrolyzed C3, and the C3B, which was the binding unit, is attached here. इससे आके आपका BB has also attached, right? So this is the attachment which has happened with the bacteria. With the bacteria, a 3B and BB, both the components are attached. Together, can I call this whole thing as 3BBB activated? I can call it as C3BBB activated, right? Now, this will convert into something which is called as C3 convertase. Now, this unit will go and activate something else. This unit will go and convert to something else. So, I am calling it a convertase. What is this convertase made up of? C3. Okay? So, I am calling this as C3 convertase. C3 convertase. Okay, for now, let's remember this much, that the C3 convertase of alternate pathway is called as C3BBB. Now, let's go back and let's look at our classical pathway. Where had we left the classical pathway? There was this bacteria, the antigen and antibody complex had formed. Then the C1 came, C1 got activated. Now, it will start the complement pathway. Who will C1 kick? Who will C1 kick? C1 will kick C4, okay. C1 will kick C4, which will kick C2. So now C1, C2, C4, everything has gotten consumed. All of these have gotten consumed. So let's consume them and let's finish it off, right. So this will kick whom? This will kick C4 and this will kick C2. C4 and C2 will both break into two segments. Yes, there is a smaller C4A and a smaller C2B, क्योंकि 2 में सब उल्टा होता है. So C4A smaller and a big C4B. Here the C2 में उल्टा होता है. C2 में bigger is what? Bigger is C2A and smaller is 2B. ठीक है? No problem. Now, where will C4A go? C4A will fly away and cause anaphylaxis. It will fly away and cause anaphylaxis. ठीक है? What will C4B and 2A do? They will attach. Both of them will attach together and they will go and bind to this guy. Right? So, ultimately what's happening? Ultimately, you have this bacteria with its lipopolysaccharide, जिससे आके आपका एंटीजन एंटीबॉडी कॉम्प्लेक्स जैसे ही बना, अल्टीमेटली इससे आके two structures have bound. What are these two structures? These are C4B and C2A. C4B and C2A have bound. Can I call this as C4B2A bracket? Now this is what is my C3 convertase. ये कौन है? ये है C3 convertase. Okay. This is C3 convertase of classical pathway. What was the C3 convertase of alternate pathway? C3BBB was the C3 convertase of alternate pathway. What is C3 convertase of classical pathway? It is C4A, so 4B2A. Alright. Now, let's look at our lectin pathway. What was happening in lectin pathway? Bacteria had come, mannose binding lectin comes. So, there is no involvement of any antibodies in lectin and alternate pathway. है ना, सीधे ही the complement is going, what is happening? Okay, some glitches happening in the chat box. Can you just see? Chat box में क्या हो रहा है? Okay, all right. So, is there any way that we can stop this comment? Can you, आप BM से बात कर लोगे ये वाला comment रुकवाने के लिए? All right. So, thank you so much for your compliments. I cannot read your name, but thank you so much. Yes, I will be covering immunology, CVS, everything from my from these lectures. चलो, क्या बोल रही थी मैं? C3b BB is the C3 convertase of your alternate pathway, and C4b2A is the C3 convertase of classical pathway. What's happening in lectin pathway? The mannose binding lectin is coming and binding directly to the mannose residues present on the bacteria. This will get activated. MBL and MASP gets activated. This will kick whom? This will again kick your C4 and your C2. Again, same thing will happen. C4 and C2. C4 will break into C4A and C4B. C2 will break into C2A and C2B, where C4 and C2A will combine. So, they will go and they will bind over here. So, what is your ultimate step? Over here, you have this bacteria. This has these mannose residues. Now, what is binding to it? C4b is binding, and here C2a is binding. तो क्या bind हुआ तुम्हारा? C4b2a active. Can I call this the C3 convertase? Ma'am, ये तो है ही C3 convertase, we have already seen, right? So, when I have to say that you have three pathways, you have classical pathway, you have lectin pathway, you have alternate pathway, and in all three, what is the starting point? The starting point of classical pathway is C1CRCS active. Yes. Starting point of lectin pathway is MBL MASP active. Yes. Starting of alternate pathway is your C3 spontaneous hydrolysis. C3 spontaneous hydrolysis. Ultimately, all three of them are converging into what? A C3 convertase formation, a C3 convertase formation, a C3 convertase formation. कहाँ चला गया? Okay, yeah. A C3 convertase formation. ठीक है ना? तीनों में ही ultimately C3 convertase बन रहा है. So all three of them are converging. So this is forming a C3 convertase, C3 convertase, C3 convertase. इसके C3 convertase का क्या नाम है? C4b2A. इसके C3 convertase का क्या नाम है? C4b2A. What is the name of the C3 convertase of this guy? C3bbb. What are all of these? All of these are C3 convertase. Correct? So when you write that the starting point of all the three complement pathways is different, but they all come together and they converge into a single C3 convertase, that is very much true right now. Yes, they have all converged into a single thing, which is C3 convertase. What will C3 convertase do? It will kickstart. कितने हो गए थे? How many enzymes we had consumed? Now C3 was pending in the middle. The C3 convertase will go and kickstart C3 now. Now C3 is also consumed. Yes. So the C3 convertase will go and it will kickstart C3. What will it do? It will break C3 into two segments. C3 convertase will break C3 into two segments. One is of course the smaller segment C3A, and one is of course the bigger segment C3B. You know what is C3A going to do? C3A is going to fly away and cause anaphylaxis. Anaphylaxis. So don't you think one of the effects of this is anaphylaxis? It will also call your phagocytes or phagocytosis. So if you have to write the final outcomes of complement pathway activation, one of the final outcomes is anaphylactic response and your cell phagocytosis. It is one of the effects. What will C3b do? B can do two things. Either this C3b will go and it will bind a pathogen. ठीक है, ये मेरा एक bacteria है, this is a bacteria. The bacteria with its cell wall and everything, fine. This C3b will go and it will totally cover this guy. This C3b has totally covered this guy and made it tasty for the phagocyte. What is this process called as? This process is called as opsonization. So either C3b can go and cause opsonization, which was one of the final outcomes which had to happen, है ना? In the first page itself we had written that one of the final outcomes is opsonization and anaphylaxis. Right at the step of C3 breaking, we have seen both the things. We have seen anaphylaxis also happening and we have seen opsonization also happening right at the step of C3b. Or since B is the binding unit, it can also continue the cascade. It can also continue the cascade for me. So what will happen when C3b decides to continue the cascade? It will go and it will bind to the C3 convertase, right? So in order to continue the cascade, it will go and bind to this guy. So what will happen now? You have your C3 convertase here. C3 convertase, it could be either of the three. Yes, it could be anything. Either it could be C4b2a or it could be C3bbb. Okay, anything. What will bind to it now? The C3b. C3b will bind to it. Now what can I call it? This, now what can be the formula of this? This can have two formula. So when C4b2a binds with C3b, can I write it as C4b2a plus 3b? है ना? If the C3 convertase is C3bbb plus 3b comes and binds, so it becomes this. So the formula can be C4b2a 3b or it can be C3bbb 3b. What are these? This C4b2a 3b and 3bbb 3b, these are your C5 convertase. What is this? This is what you call as the C5 convertase. C5 convertase. Now the C5 convertase will also do what? It will kick C5. When it will kick C5, either it will break into a small C5a, which will go and do the same thing, anaphylaxis and cell phagocytosis, or it will break into C5b, which is the bigger and the binding fragment. ठीक है? So अब चलते हैं अपने कौन से वाले पे, अपने सारे देख लेते हैं कंज्यूम हुए नहीं हुए. C1 हो गया यूज, हो गया क्लासिकल पाथवे में. C2 हो गया यूज, यस क्लासिकल ऑल्टरनेट दोनों में. C3 हो गया यूज, यस इट हैज बीन यूज्ड इन C3 कन्वर्टेज. C4 इज यूज्ड, यस. C5 is used, yes, C5 convertase has broken C5. Now this C5 will bind everything. It will bind C6, C7, C8, C9. All of the enzymes are going to get consumed now. So what is this C5b doing? C5b will go and bind C6, C7, C8 and C9. So what is this? C6, C7, C8, C9. Everything has been bound together. Now what is this? This is like the bullets of a machine gun. This will create holes inside the bacterium. So if this is my bacteria, this complex is going to create holes. Is going to create these poles pores or holes inside this bacterium. What is this called as? This is called as MAC or membrane attack complex formation. So what was the final step which had to happen out of this complement activation? MAC formation. And we have just seen how this MAC formation happens. So what is the formula of MAC or membrane attack complex? C5b2c9. है ना? If you have to write a short form, what is MAC? MAC is C5b2c9 complex. That is what is MAC. Now I don't think any doubt remains in the understanding of complement pathway and complement system, and I hope that now you will pray that they in fact give you complement system in the exam. ठीक है ना? It is done and it is dusted. So we're done with complement, a very complex but a very high yield but a very simple topic. Simply remember the pathway. You know the starting point is antigen antibody complex formation, or the starting point is C1 activation, which is antigen antibody complex formation, or the starting point is mannose binding, or the starting point is C3 hydrolysis. किसी तरह C3 convertase पे पहुंचेंगे, which is C4b2a or C3bb. It will combine with 5b form C5 convertase, which will make C5b bind to C6, 7, 8, 9, making the membrane attack complex. You are welcome, guys. You are welcome. All right. Now, another differences table. अब तक सबको समझ आ गया humoral immunity and cell-mediated immunity, है ना? You know it. You know that humoral immunity basically means antibody mediated immunity. एक बार just to give you थोड़ा सा will power and strength, let's realize that we have converted primary, secondary, active, passive, complement. Quickly we are going to go over the table of CMI and humoral, and very quickly we are going to come on to MHC, है ना? बहुत जल्दी से हम अपने मेजर हिस्टोकम्पैटिबिलिटी कॉम्प्लेक्स के टॉपिक पे आ जाएंगे, सो डोंट वरी, एंड देन वी विल हैव अ मच क्लैरिटी ऑल्सो ओवर दैट टॉपिक. ठीक है, फिर हम क्विकली हाइपर सेंसिटिविटी भी खत्म कर लेंगे एंड देन विल बी डन. चलो, लाइक आई सेड, ह्यूमोरल वर्सेस cell mediated. So stay with me, this is not the time to get tired. No thinking that I will watch it later. No, watch it right now and get it done with. That is the purpose of FARRE, that is the purpose of revision, that with me you revise and you get it done with. Okay, so no leaving right now at this point of time, no coming back later, no watching the lecture later. Watch it right now and finish it off. That is how revision is done. Revision is not done later, revision is done now. Alright, chalo. So kya bola? Humoral and cell mediated immunity. Humoral immunity is associated with B lymphocytes and it is responsible for destroying pathogens by producing antibodies against them. You know it simply that humoral immunity is antibody mediated and cell mediated immunity is T cell mediated. ठीक है, B cell and T cell. B cells क्या हैं? These are the immune cells of the lymphocyte lineage which take birth in the bone marrow. So I always say bone marrow is like the mother of all organs. वो माँ है. Everybody gets born there. So the entire cellular lineage, the blood cell lineage. Alright, Aditi, there are detailed batches which are detailed lectures batch launch. So detailed batches are the Nishchay batches which are already launched. But yes, point noted. Let's see what we can do about this. चलो, क्या बोला हमने? Humoral immunity and हाँ, क्या बोल रही थी मैं कि bone marrow is the mother of all organs, है ना? क्या है वो? जहाँ पे सारे आपके cellular cells of the blood they take birth. So the B lymphocyte lineage, T lymphocyte lineage, erythroblast, सब कुछ bone marrow में gets born. So these B cells also get born in the bone marrow, the T cells also get born in the bone marrow. Now they have to mature, they have to go to college. They will choose to either go to college in the bone marrow itself. Jैसे many of you have chosen the MBBS college in your home state itself. Many of you have chosen to go to college in the in a separate city. So the cells which decide to go to college in thymus in a separate city are the T cells. So both B cells and T cells originate where do they originate? They originate in the bone marrow itself. But where do they go to mature? B cells mature within the bone marrow itself and T cells go to mature in the thymus. Alright. So humoral immunity is mediated by the B lymphocytes which produce antibodies, and cell mediated is by the T lymphocytes. You know the T lymphocytes can be T helper cells and the cytotoxic T cells. Also cell mediated immunity is by the natural killer cells and macrophages. So one thing is very important to understand, that humoral immunity is a component of adaptive immune response, guys, acquired immune response, because antibodies are produced in response to adaptive immunity, whereas the T cell or the cell mediated immunity is a part of your natural or innate ability also, because innate immunity ke cells hai macrophages and natural killer cells. They are also cells. What are they doing? They are going and causing killing of the pathogen. So that is also cell mediated. So now humoral is only B lymphocyte mediated, whereas cell mediated is T cell, both T helper and cytotoxic T, as well as natural killer cell, as well as your macrophages. Antibodies किसमें involved है? Antibodies are involved in the humoral immunity and they are not, absolutely not involved in your cell mediated immunity. Cell mediated immunity is totally cell mediated. वहाँ cells काम कर रहे हैं. फंक्शन क्या है? ह्यूमोरल इम्यूनिटी एंटीजन को रिकॉग्नाइज करेगा और उसकी जगह एंटीबॉडीज बनाएगा. एंड ह्यूमोरल इम्यूनिटी इज नोन फॉर वर्किंग अगेंस्ट एक्स्ट्रा सेल्युलर पैथोजेंस, बिकॉज व्हाट डू दीज एंटीबॉडीज डू? दे फाइंड अ फ्री मूविंग ऑर्गेनिज्म, दे बाइंड टू इट एंड दे न्यूट्रलाइज इट. ठीक है, that is what the antibodies do. They bind to it and they neutralize the pathogen. What do cells do? What is the cell-mediated immunity do? Here, it works against intracellular pathogens. Intracellular pathogens also. So, you have many viruses which are obligate intracellular. है ना? Viruses तो सारे ही intracellular हैं. They cannot survive outside the host cell. They are only present inside the cells of the body. Then you have certain bacteria which are intracellular. Then you have certain bacteria and fungus which does not live inside your cell. They live outside your cell, right? So, anything which is outside your cell freely living, antibodies will attack it. Anything which is hiding inside your own body cell, there these intracellular pathogens can only get killed by the cell mediated immunity or these cells of the cell mediated immunity. ये कैसे करेंगे? They are related to T lymphocytes which work by identifying viruses or any microorganisms, destroying them through cell lysis, phagocytosis. ठीक है? So they are going to directly go and kill these cells. Here antibodies are secreted, here cytokines are secreted. What are cytokines? These are these enzymes or chemicals which are released by the cells in order to activate other cells. ठीक है? तो whenever a T helper cell gets activated, it releases interleukins, it releases tissue necrosis factor, it releases many cytokines. These cytokines go and activate macrophages, they activate killer cells, they also activate antibody producing machinery also sometimes. But the cell mediated immunity, all the information in the cell mediated immunity is by the cytokines, whereas in the humoral immunity the antibodies, they act directly. Here it is rapid or quick response. Humoral immunity is quick, whereas cell mediated immunity is slow or it is delayed. Humoral immunity, now we are also going to cover hypersensitivity. So in that hypersensitivity type 1, 2 and 3 are your humoral immunity. The only hypersensitivity type which is hypersensitivity type 4 is cell mediated immunity. ठीक है? The only type 4 is a cell mediated immunity. वहीं पे रिजेक्शंस का क्या मतलब कि जो आपके ग्राफ्ट रिजेक्शंस होते हैं, ह्यूमोरल इम्यूनिटी इज इन्वॉल्व इन अर्ली स्टेज ऑफ ग्राफ्ट रिजेक्शन, जैसे अगर प्री फॉर्म्ड एंटीबॉडीज हैं किसी के ब्लड में, वेयर एज सेल मीडिएटेड इम्यूनिटी इज इन्वॉल्व इन दी प्रॉपर रिजेक्शन ऑफ द ऑर्गन ट्रांसप्लांट्स. ठीक है? सो दिस इज एन एक्स्ट्रा स्टेप दैट यू कैन ऐड that people who are already sensitized, they have antibodies already in them. So if they take a transplant or a graft, Immediate rejection happens because of humoral immunity or antibodies. Whereas the rejection which happens over time once the organ is implanted, then the patient is fine in the hospital, patient is discharged, goes home. Then if the graft gets rejected, that is the cell-mediated immunity playing its role, right? So you can write these differences between humoral and cell-mediated. You will write the meaning, you will tell the most important cells associated, you will tell whether antibodies are associated with them, what is secreted by these cells, how do they act against these pathogens, in which hypersensitivity, which response, immune response is associated, whether humoral or cell mediated. Now I talked so much about T cell, B cell, going to college here and there. So sometimes they also ask you to write differences between T cells and B cells. So what are T cells? Where do they originate? Both of them will originate in the bone marrow itself. Okay? Bone marrow is the ultimate mother organ of all. That is where all the cells they originate. Now, where do the T cells go to mature? Cells go to mature in thymus, that is why they are called as T. T for thymus, T for T cells. B for bone marrow, B for B cells. So wherever the cell goes to mature, wherever place the cell goes to get into college, है ना? यही होता है ना, you go to Harding, you are a Hardonian, you go to Ames, you are an Amesonian. So similarly, the cells also get their name. They go to thymus to mature, they are T cells. They stay back in bone marrow to mature, they are called as B cells. Now T cells are going to have some antigen recognition receptors. हर cell के ऊपर they have the receptors which will identify or recognize certain antigens. Yes, so the T cells have the CD3 receptors on them. This is a marker of a T cell. A T cell is a T cell when it expresses the CD3, whereas B cells have IgM or IgD on top of them. They also have CD4 and CD8 markers which are going to recognize these pathogens. ठीक है, तो क्या होता है किसी भी B cell के पास और क्या होता है किसी भी T cell के पास. If this is a B cell, even though all of this has been covered in the one shot, I am just repeating it. It will have certain receptors. What are these? These are called as B cell receptors. B cell receptors, है ना? BCR. Now what is this? This is a marker of this cell. Whenever a B cell receptor is present on a cell, then I will say that this is a B cell. This is also a receptor to which an antigen is going to come and bind. So there are dual roles of the B cell receptor. A, it will identify the antigen, and B, whenever I look at a cell and I see that the cell has a B cell receptor, then I know that this is a B cell. ठीक है? Similarly, you have the T cells. A T cell is a T cell because it has the CD3 receptor on top of it. When this T cell matures, it can become of two types. Either it can have, so both of them are definitely going to have a CD3, no doubt there. Then, depending upon which T cell it is, either it can make a CD4 receptor or it can develop a CD8 receptor. If it is CD4, 4 looks like H, so that's a T helper cell. If it's a CD8, I always tell you, remember I8, 8 means phagocytose, 8 means to kill. So this is the cytotoxic T cell or the killer T cell, right? So these are all the receptors which are present on the surfaces of these cells. If I look at a cell and I see the CD3 marker, I know it's a T cell. When I look at a cell, I see CD3 plus C4, I know it's a T helper. I look at CD3 plus C8, I know it's a cytotoxic T cell. What is the function of CD4 and CD8? They also bind to the antigen. They also bind to the antigen. Similarly, I look at a cell, I see B cell receptor, I know it's a B cell. What is the function of B cell receptor? To bind to the antigen. What is the nature of this B cell receptor? Do you know that these B cell receptors by themselves are antibodies? Okay, a B cell after activation eventually produces antibody, correct? But the receptor which is present on a B cell surface, which makes a B cell is also an antibody, specific antibody, which either IgM or IgD, no other antibody. After activation, a B cell can produce all different 5 classes of antibody, A, B, C, D, E. But before activation, that is the receptor on the B cell, which will bind to the antigen is always a IgM or an IgD, which is a B cell receptor. Okay, which is a B cell receptor. Alright, so same thing is written over there. What is the CD marker? C3, 4 and 8. What is the receptor? CD3 in a T cell. IgM or IgD is the receptor on the B cell. It also has some CD molecules such as CD19, 21 or 24. Thymus specific antigens are present in the T cells, they are absent in the B cells. Why? Because it is only the T cell which has gone and matured in the thymus, the B cell has not matured in the thymus. That is why thymus specific antigen is only present inside a T cell. Okay? Who is going to have the American accent? Only the people who have lived in America, not the people who have lived in Goregaon, right? So that is why thymus specific antigen is only present in the cells which have matured in the thymus. That is why thymus specific antigen is only present in the T cell. Then you have microvilli on the surface. Iska kya matlab hai? Microvilli on the surface basically means that these small finger like projections. So the B cells have these small finger like projections because that increases the surface area, right? So just look at this. If this B cell receptor is present on the surface of a B cell, how many can it accommodate? A sphere can only accommodate a limited number of B cells. But what if this guy has a shape like this? Okay, if the shape is like this, then in each and every area these B cell receptors can be there. So that increases the number of B cell receptors by a lot. ठीक है, बहुत सारे B cell receptor अब इसके अंदर accommodate हो सकते हैं. That is why a B cell has all of these microvilli coming out on its surface projecting, because that can accommodate a lot more B cell receptors. So these microvilli are present. They are absent in the T cell, but they are very much present in the B cells. Then you have antibody recognition receptors. None. A T cell does not need to recognize an antibody, whereas B cell receptors itself are the IgM or the IgD, like I had told you, right? So you can easily write a differences table because it is always a problem to come up with what to write, but now you can easily write it. Alright guys, so those who are staying, congratulations, good job. You're revising at a great pace. You're going to have all your queries solved and answered. Now we move on to the most controversial, the most important topic, which is MHC or major histocompatibility complex classes. ठीक है? Class 1 and class 2 of the major histocompatibility complexes. So no getting tired right now, because I think we'll just take a few moments, a few minutes, and quickly we will finish off a complex topic, right? If you sit down and read from the book, you might forget it, but I am going to explain it to you simply and very well. देखो, MHC stands for Major Histocompatibility Complex. Anybody with a basic understanding of English can understand. Major, it is quite major. Histocompatibility, meaning compatibility between the cells. If I am speaking and you are listening and you don't understand a word I am saying, there is no compatibility. If I am speaking and you are listening and you understand everything that I am saying, there is excellent compatibility. Yes? So two cells can either be compatible, they can be non-compatible. There are certain set of genes which create certain proteins which determine whether two cells are compatible with each other or not. For a person to stay stable, compatibility is needed. All the cells should not start fighting with each other. That is why whenever I say, or anybody says in this world that you are unique, there is nobody like you in this world, there is actual scientific backup reasoning for that. What is the reason? MHC. Everybody is unique because all of our cells have specific proteins which are only and only specific to us, ourselves. ठीक है? These guys are only specific specially to me. What are they? MHC. MHC is nothing but it's a group of genes. It's a group of genes. MHC is not the protein. MHC codes for the proteins which are also called as MHC, but it is a group of genes. So why is it a complex? Because it is a group. What do these genes do? Every gene will code for something. Mostly the genes, what do? They translate into proteins. है ना? So these genes are going to code for a set of host cell surface molecules. क्या मतलब हुआ set of host cell surface molecules? Receptor, है ना? Host के cell पे कोई भी molecule है, what is that? That is a receptor. That is the thing which is going to attach to something, right? So the meaning of this big word is receptor. Let's do something. Let's try to draw what they are saying. They are saying that MHC is a group of genes which codes for certain receptors on the top of a cell. So suppose this is a cell of my body and it is saying that it is coding for a certain receptor on its surface. What is this? This is the receptor which has been coded by the major histocompatibility complex. So the MHC is coding for this receptor on this cell. ठीक है? Okay. Now what will this receptor do? This receptor binds to the peptide fragments derived from a pathogen. इसका क्या मीनिंग है पेप्टाइड फ्रेगमेंट डिराइव्ड फ्रॉम अ पैथोजन? एंटीजन, है ना? एंटीजन इज प्रेजेंट ऑन सरफेस ऑफ अ पैथोजन. So that peptide fragment on the surface of the pathogen is called as an antigen. So what is the meaning of this line? This line simply means antigen. So up till now this is just saying that major histocompatibility complex is a group of genes which codes for certain receptors which are present on the host cell, and these receptors, they bind to a particular antigen. They bind to a particular antigen. ठीक है? Okay. And display them on the host cell surface. यही तो हुआ है. It has bound to this antigen and displayed it on its own surface. So I am the cell. Here is a receptor. My hands are the receptors. These receptors have bound to an antigen and now I am showing you this antigen. This is what is happening. This antigen is being displayed on my surface. Yes, I am the host cell. These are my MHCs. I am holding antigens and I am showing them to you. This is what is happening. All right. And it is saying for recognition by appropriate T cells. So why is this cell displaying it on its cell surface? Because this cell is going to display it to a T cell. So can I call this cell as a cell which is presenting the antigen? So can I call this cell as an antigen presenting cell? So in the third line, this definition has told me that this host cell is nothing but an antigen presenting cell. ठीक है? So what is this host cell? This host cell is nothing but an antigen presenting cell. And it is saying that it is displaying this antigen on its surface for processing by a particular T cell. इसने बोला कि this is now going to get recognized by a particular T cell. Doesn't this diagram ring a bell in your mind? Isn't this the basis of T cell activation? We have read this so many times in so many different lectures, also covered in the lecture on the one shot, also covered in live classes. So many times we know that how does the T cell activation happen? A T cell cannot get activated on its own. A T cell needs an antigen presenting cell to get activated. I have told you depending upon which CD marker is this. What is this now? This is a receptor of the T cell, so this is a T cell receptor. You know that a T cell receptor can either be a CD4 or a CD8. Can either be a CD4 or a CD8. Yeah, na? If it is a CD4, this T cell is a T helper cell because 4 looks like H. If it is a CD8, it is going to ate the cell, meaning it is going to kill the cell. It is a cytotoxic T cell, right? And you know that always a T cell for activation needs an antigen presenting cell to present the antigen. This MHC molecule is of 2 types and you all remember the rule of 8. Rule of 8. What does the rule of 8 tell me? That the multiplication of which MHC has brought the antigen and which CD4 it is binding to should be 8. So dash into 4 is 8 and dash into 8 is 8. है ना? ये CD4 है, ये CD8 है. So dash into 4 is 8, 2 into 4 and 1 into 8. So don't you think The MHC which binds to CD4 is MHC1 and the MHC2 and the MHC which binds to CD8 is MHC1, है ना? ये हम कितनी बार कर चुके हैं? That is exactly what the definition is telling me. This diagram I have made before when I read T cell activation in the cell-mediated immunity. The same diagram I am making again to explain that what are major histocompatibility complexes. They are nothing but they are a group of genes which are coding for these proteins which are present on the antigen presenting cells. ठीक है? What do the antigen presenting cells do? They present an antigen and they show it to a T cell. Now that permutation combination we have revised again, right? अच्छा जी, मैंने क्या बोला? What are MHCs? They are a group of receptors present on APCs. Something must be coding for them. The MHC genes, complex of MHC genes. Now आपने बहुत सारी जगह पढ़ा होगा HLA. Something which is called as HLA. What is the difference between an MHC and an HLA? Right? What is the difference? So many places you hear the word HLA written. Is it something different or it means the same thing? I had said that this MHC molecule is present on any antigen presenting cell. What are these antigen presenting cells? Which cells of my body are antigen presenting cells? You know it that examples of antigen presenting cells are macrophages, dendritic cells, and also your B cells sometimes can also behave like antigen presenting cells. Even though B cells are totally different and they are a part of antibody production, sometimes they themselves can also act like antigen presenting cells. These APCs, they have MHC2 molecules on them, ठीक है? Whereas MHC1 is present on all the cells of my body. So MHC is actually present in almost all cells of the human body except sperm cells. ठीक है? So where is MHC class 1 present? On all the nucleated cells of the body. So MHC is not present on RBCs, MHC is not present on the sperm cells. But MHC overall, 1 and 2 combined, MHC 1 and 2 combined is present in almost all cells of the body except sperm cells and RBCs. But they were first discovered on the surface of the leukocytes. So then we started calling them human leukocyte antigen. But later on we discovered that they are not only present on the leukocyte, they are present in almost all cells of the body. So later on we just started calling them major histocompatibility complexes or MHC proteins which are coded by the MHC genes. Okay, so HLA and MHC is one and the same thing. It's just that these MHC genes were first discovered on the surface of leukocytes. So we started calling them human leukocyte antigen. Otherwise it means the one and the same thing. Okay, so what happens? There is a chromosome. The chromosome has little surfaces. Those little surfaces have gene clusters. These gene clusters code for a set of genes. Okay, these set of genes can be HLA, A, B, C. A can also be HLA, A1, A2, A3, A4. Then it could be HLA, D. HLA, D can be HLA, DR, DQ, DP. सब कुछ क्या है? This is nothing but a set of genes. Right? These set of genes will make up the proteins. है ना? यही तो बोल रहे हैं हम. MHC क्या है? ये genes हैं जो कि कुछ रिसेप्टर के लिए कोड कर रहे हैं. जीन का नाम भी MHC है, रिसेप्टर का नाम भी MHC है. ठीक है, इसलिए कंफ्यूजन हो जाता है हमें. सो कुछ जीन्स हैं जो कि इस MHC रिसेप्टर को कोड कर रहे हैं. तो कंफ्यूजन हटाने के लिए हम क्या कर लेते हैं कि हम जीन को HLA बुला लेते हैं और उससे कोड होने वाले रिसेप्टर को MHC ही बोलते रहते हैं, है ना. सो हमने क्या किया, हमने MHC कोडेड प्रोटीन्स जिनको ल्यूकोसाइट पे देखा तो HLA बोला और बाकी जो प्रोटीन्स हैं उनको MHC बोला. दो तरह की जीन क्लस्टर्स हैं. ठीक है, हर एक जीन क्लस्टर, जैसे एक जीन क्लस्टर है वो MHC1 बना रहा है, एक जीन क्लस्टर है जो MHC2 बना रहा है. ठीक है, बात समझ में आई. सो जब हम MHC1 या 2 को डिफ्रेंशिएट करते हैं तो हमें पता है कि जो MHC क्लास 1 है, वी नो दैट MHC क्लास 1 is present on all nucleated cells except sperms, and it is also present on the platelets. Whereas MHC class 2 is only present on the antigen presenting cells, right? The class 1 presents the peptide antigen to the CD8 T cells, because 1 into 8 is 8. And the MHC class 2 presents the antigen to the CD4 T cells, because 2 into 4 is 8. We have just seen it. The nature of peptide antigen, the nature of antigen which this MHC shows to the T helper, to the T cell, is MHC class 1 will take endogenous or intracellular antigens. ठीक है? Whereas MHC class 2 will take exogenous antigens and present to the T cells. The MHC class 1 का peptide binding site is present where? It is present in the alpha 1 alpha 2 groove, and it is present in alpha 1 beta 1 groove. अब ये क्या बोल दिया? Right? अब ये क्या बोल दिया हमने? देखो क्या होता है. This is a cell. This is a cell. उसकी ये सेल मेम्ब्रेन है. इस सेल मेम्ब्रेन के ऊपर जो MHC मॉलिक्यूल या HLA मॉलिक्यूल है उसका नाम है MHC class 1. ये है MHC class 2. They are made up of alpha and beta chains. So let's imagine that this is what? This is all the nucleated cells. All the nucleated cells. And let's imagine that this here is a antigen presenting cell. ठीक है? So the MHC molecule which is going to be on all nucleated cells is MHC1. The MHC molecule on APC is going to be MHC2. You know what is MHC1 made up of? It is made up of alpha and beta units. क्या होता है? It has 3 alpha units. 3 alpha units. So you have an alpha 1, then you have an alpha 2, then you have an alpha 3. They are attached like this. Now don't you think that this will fall on itself if it is unsupported? So it gets a support from a beta 2 microglobulin. Okay, from a beta 2 microglobulin. So what is happening here? You have alpha 1, alpha 2, alpha 3, and here a beta 2 has come. Okay, what? This is what? This is MHC class 1. Yes, all right. Now, okay, what is happening in APC? Here also alpha beta is there, but this is alpha 1 alpha 2, and it has another one beta 2. Simple. So what are these? Alpha 1 alpha 2, beta 1 beta 2. Now if an antigen has to come and bind, MHC class 2 is quite simple. MHC 2 is quite simple, right? Antigen will come and bind over here. So where is the antigen binding? In the groove which is present in alpha 1 and beta 1, है ना? यहाँ पे एक ऐसे groove बना था, इस groove के अंदर ये antigen आके बैठ गया. Here, where is the groove? Here the groove is here, है ना? This is the groove in which the antigen will sit. So what is the groove of MHC1? Alpha 1 alpha 2 groove is where the antigen sits. And in MHC2, alpha 1 beta 1 groove is where the antigen sits, right? So what was the last point of difference? That the peptide binding site, meaning antigen binding site, is alpha 1 alpha 2 groove in MHC class 1, and it is alpha 1 beta 1 groove in MHC class 2. Alright, so I hope everything is now clear to you. Whatever I have said is now clear to you. If any doubts, please let me know. Otherwise, we can move on. Otherwise, we can move on to the last topic, which is hypersensitivity. Hypersensitivity. So, we are done with CMI, we are done with MHC, and lastly, we are going to get done with your hypersensitivity reaction. Excuse me. एक मिनट ना. इसको लगा देना. खोल लो. वो देख लेना लूज ना हो, राइट गाइज। थर्ड पॉइंट का मतलब बेटा MHC के डिफरेंसेस का थर्ड पॉइंट, nature of peptide antigen. Basically this is simply telling you that which type of an antigen the MHC molecule likes to bind to. ठीक है, तो यूजुअली जो आपके MHC क्लास 1 वाले सेल्स होते हैं, MHC 1 पे आपके यूजुअली इंट्रासेल्युलर पैथोजेंस आर प्रेजेंटेड, right? So intracellular pathogens are like viruses and also certain tumor antigens. They are endogenous or they are present, they usually live inside the cell, inside the host cell, है ना? उसमें लाइट जलेगी जब ऑन होगा, right? So usually these intracellular type of pathogens, they are presented by MHC class 1 molecules. And your exogenous type, matlab who are not obligate intracellular pathogens, they are presented by the MHC class 2 type of antigen presenting cells. That is the only point that they are telling. Alright, so are we ready to begin hypersensitivity reactions? Yes, so the same thing was being shown by this diagram. Here you had the MHC class 1 with the alpha 1, alpha 2, alpha 3 and the beta 2 microglobulin balancing the alpha chain, and then in the groove of alpha 1 and alpha 2 you had the antigen binding. Over here you had alpha 1, alpha 2, beta 1, beta 2, and in the groove of alpha 1 and beta 1 you had the antigen binding site. Alright, got it ma'am, theek hai, done. So now we are talking about hypersensitivity reactions. So what is a hypersensitivity reaction? A hypersensitivity reaction is an exaggerated response. Okay? That is what you have to write majorly. What is a hypersensitivity reaction? The first thing which is important in the definition, that it's an exaggerated immune response. मतलब कि अब ज़्यादा ही कुछ over react कर रही है body. Yes? The immune response or the immune system is just behaving like too much, है ना, overreaction. Another important prerequisite is that the host must be sensitized. पहले से चोट खाया होना चाहिए. The host must be sensitized. What is the meaning of sensitized? Meaning that the host has already seen that allergen or that pathogen before, right? Now there is a secondary attack, and in this secondary attack there is so much of exaggerated immune response that it is no longer only damaging the pathogen, it is in fact damaging the host itself, right? So what happens, that there is a secondary or repeat exposure, okay, to the antigen. कौन एक्सपोज हुआ है? एक sensitized host. Secondary or repeat exposure to antigen of the or by the sensitized host. Okay? And this sensitized host has generated an immune response which is an exaggerated immune response. So this is an hypersensitivity reaction, is an exaggerated immune response to a secondary or repeat exposure by to an antigen by a sensitized host. ठीक है ना, एक होस्ट था जो कि सेंसिटाइज था और फिर उसने फिर से एक दोबारा से एंटीजन को देखा, लेकिन वो और फिर उसने एक हाइपर एग्जैजरेटेड रिस्पॉन्स दिया. सो यू हैव दिस क्लासिफिकेशन फॉर हाइपर सेंसिटिविटी रिएक्शन कॉल्ड एज जेल एंड कूम्स क्लासिफिकेशन. Gel and Coombs classification. What does Gel and Coombs classification say? It says that there are 4 types of hypersensitivity reactions. Type 1, type 2, type 3 and type 4, right? So you have hypersensitivity reaction type 1, hypersensitivity reaction type 2, hypersensitivity reaction type 3 and hypersensitivity reaction type 4. Now I have a set of mnemonics for you. How to remember all of this? हमने 1 2 3 we have written 1 2 3 4. Can we also write it as A B C D? So I am writing A B C and D. So simply remember 1 2 3 4 as A B C D. Okay. So what is A? A stands for allergy. So actually type 1 hypersensitivity reaction is allergy or allergic response. What is B? B means antibody. Antibody. So what is type 2? Type 2 is antibody mediated hypersensitivity reaction. Right? What is C? C stands for complex. So which complexes are formed in the body? Antigen antibody complex. So type 3 reaction is antigen antibody complex mediated hypersensitivity reaction. And what is D? D stands for delayed type hypersensitivity. Delayed type, also called as DTH, delayed type hypersensitivity. ठीक है? I have another mnemonic to remember the mechanism of reactions. ठीक है? कैसे होंगे? What is the mechanism of these hypersensitivity reactions? See, allergy, the person who has the ability to generate this exaggerated allergy is called as an atopic individual. So my genetic predisposition to have an allergic response is called as atopy, right? When this allergic response includes all the systems of my body, that is called as anaphylaxis, right? That is called as anaphylaxis. You know which major antibody is responsible for allergy? I always tell you the mnemonic, whenever you are allergic to something, you just say U, right? So U is E, IgE types. So what is the main antibody involved in type 1 hypersensitivity reaction? It is IgE mediated, right? So type 1 hypersensitivity reaction is allergy, which is IgE mediated, all right? Now you have antibody. What does antibody do? Antibody causes cell lysis, that is cytotoxic. So this is antibody mediated response, which is cytotoxic response. Antibody is cytotoxic. Let me do something. Instead of cytotoxic, let's write it as cytotoxic. Cytotoxic. So this can help me remember that type 2 response is antibody mediated cytotoxicity. What is it? Antibody mediated cytotoxicity. So कौन सी antibodies involved होंगी? Obviously IgE is involved here. So in this type 2 you have the IgG or the IgM getting involved, and what are they going to lead to? Because of this cytotoxic nature, they are going to lead to antibody dependent cellular cytotoxicity. We will just see what is this antibody dependent cellular cytotoxicity, because that itself is a short note also. Okay, ADCC. So what are the main mechanisms of type 2 hypersensitivity? Antibody mediated lysis and antibody dependent cellular cytotoxicity. So IgG or IgM mediated neutralization, or there is ADCC formation, or of course if antigen antibody is forming, the antibodies are acting, so they can also take help of the complement system. कोई बात नहीं, right? Done. What is type 3? Type 3 is C. So you know, free rhymes with 3, right? Free rhymes with 3. So these antigen antibody complexes are the free complexes. They are freely floating antigen antibody complexes which settle in different sites of the body and cause damage. Okay? So there is an antigen and the antibody, both have formed a complex. They are now supposed to activate the classical complement pathway and die off. But nothing has happened. They are freely floating about, and then they go to maybe a kidney or to a bone, and then they just settle there. Now all of the immune system gets activated, tries to damage this complex. As a turn, damages the area surrounding that complex, leading to glomerulonephritis, leading to SLE. Yes, this free antigen. So what is the mechanism of type 3? It is free antigen antibody complex deposition in different sites or parts of the body. That is type 3 hypersensitivity reaction. What is type 4? Type 4 is delayed type hypersensitivity, and it is the only thing which is actually a cell-mediated immune response. Don't you think here an antibody is responsible, so it is a humoral immune response? Here antibodies are responsible, it is a humoral immune response. Here also antigen antibody complexes are formed, humoral immune response. So the three, type 1, 2 and 3 are actually your humoral immune responses. ये हमने, this we have seen in the differences table between humoral and cell-mediated also. And the only cell-mediated immunity hypersensitivity type is the delayed type hypersensitivity or type 4 hypersensitivity reaction. ठीक है? Now, one kind of looks like I, right? One looks like I. So, type 1 hypersensitivity happens immediately within a few seconds. Whereas delayed type is delayed, so it will take almost 2 to 3 days to develop. Yes, so type 1 is immediate, 1 looks like I, it is immediate, whereas type 2 and 3 takes some few minutes to a few hours, and type 4 takes around 2 to 3 days to develop. ठीक है? Like I said, type 1, 2, 3 all are humoral immune responses, and type 4 is a cell-mediated immune response. Time taken, type 1 happens within minutes to hours immediately, type 2 and 3 take a few hours, whereas type 4 takes around 2 to 3 days, it is delayed, right? The mediator of type 1 is the IgE antibody, type 2 is the cell surface bound IgG and IgM leading to antibody dependent cellular cytotoxicity or ADCC. Type 3 is the antigen antibody complex, and this is cell mediated, so that is why only your T cells are involved in the type 4 hypersensitivity. Effector mechanism क्या है? यहाँ पे mast cell degranulation होता है which ultimately leads to anaphylaxis. So now we have to see the process how mast cell degranulation is happening. Type 2 में there is ADCC and complement mediated cytolysis. So we have to now see how does ADCC happen. So now what all do we have to read? We have to read mast cell degranulation in type 1 hypersensitivity. We have to read ADCC in type 2 hypersensitivity. ये simple है. Antigen antibody complex deposits in various organs and initiate an immune response. That immune response which has come to kill this antigen antibody which has settled in the organ ends up killing the other cells of the organ also. So leading to damage. Imagine there was a free antigen antibody complex floating about, goes to the kidney, settles in the basement membrane. Now the immune cells come trying to kill this complex, end up killing the glomerular basement membrane, leading to post-streptococcal glomerulonephritis. Similarly, end up damaging the, giving the rashes on the skin, leading to SLE. Okay, so all of these are examples of your type 3. Serum sickness, Arthus reaction, glomerulonephritis, क्या है ये सब? Type 3 hypersensitivity reaction. Quite easy, quite simple. Example is SLE. It has already been done with the example that I just told you. Type 4 is a CD8 cell causes macrophage activation which leads to phagocytosis. So we have to see how the type 4 reaction also happens, right? So quickly we will start our discussion with the type 1 hypersensitivity reaction. है ना? क्या है आपका? Type 1 hypersensitivity reaction which is nothing but your allergy. Now I have one mnemonic. Right now we are going to summon Anil Kapoor. What did Anil Kapoor sing? 1 2 ka 4, 4 2 ka 1, my name is Lakhan, my name is Lakhan. Yes, so that is our mnemonic. What is the mnemonic? 1 2 ka 4. ये 1 2 ka 4 is also the mnemonic that we have read for T cell activation. Those who have watched the one shot lecture, they will know that T cell activation में easy था 1 2 ka 4. If it is T helper 1, it releases interleukin 2. If it is T helper 2, it releases interleukin 4. है ना? All of this we know. Interleukin 4 goes and says 4 बातें in the ear of a B cell. So interleukin 4 activates a B cell and interleukin 2 activates a cytotoxic T cell. You know all of this, ठीक है? छोड़ो. Now we have to focus on hypersensitivity reaction. So let's forget about T cell B cell activation. Let's only focus on hypersensitivity reaction 1. What did I say? An allergen will come. So this is an allergen, this is a pollen grain, this is anything, this is an irritating allergen. First of all, what is the prerequisite for a hypersensitivity reaction? That it has to be happen in a sensitized host. So the first step of type 1 hypersensitivity is going to be host sensitization, right? So first we have to do host sensitization. So what is the first step we are doing? We are doing host sensitization. So first of all, for the first time in my life, primarily an allergen comes. This allergen is going to be seen by an antigen presenting cell, right? Antigen presenting cell is going to internalize it. It will show it to a T cell. क्या करता है antigen presenting cell? अपने bicep या MHC पे रख के इस antigen को it shows or presents to any T cell. Yes, to any T cell. Now, which T cell? A T helper cell. Which T helper cell? T helper 1 or T helper 2? How to remember this? What is the mnemonic 1, 2 ka 4? So, if the hypersensitivity reaction is 1, which MHC is going to, which T helper is going to be included? 2. Okay. So, what is it? T helper 2 cell. You know T helper 2 has what? It has CD4 and you know 4 into 2 is 8. So, it has to be MHC 2. So, there is no scope of confusion anymore. Everything is interlinked, okay. So, APC has come, it has presented the antigen, my allergen, to a T helper 2 cell. What will the T helper 2 release 1, 2 ka 4? It will release interleukin 4. So, T helper 2 has now released interleukin 4. Which interleukin? 1, 2 ka 4? Interleukin 4. ठीक है? Interleukin 4 will go and speak 4 बातें. It will say 4 बातें B cell के कान में कहेगा. Okay? Interleukin 4 will go and activate B cell. B cell. What will B cell do? B cell will activate into a plasma cell. Plasma cell will produce antibodies. What are these antibodies? IgE antibodies. What are these? These are IgE antibodies which are specific against this allergen, right? Specific antibodies against the allergen have been produced. This is the IgE. You know, there's another guy who's floating about in the body at this time. His name is a mast cell. Who is a mast cell? It's a mast cell. Okay? So, there is a mast cell or a mast cell which was floating about, and a mast cell is a granular cell. Granular cell, right? This mast cell has some important receptors on its surface. You know what are these receptors? These receptors love IgE antibodies. These receptors love IgE antibodies. What are these black guys? These are mast cell receptors, receptors of the mast cell. They love these IgE antibodies. So, these IgE which has been formed will bind to this from their Fc portion. I hope you know this, that every antibody has a Fab portion which is the antigen binding portion and an Fc portion or the constant portion. And what do these antibodies do? They bind from this Fc portion to the cells of my body. They can bind to a mast cell, they can bind to a natural killer cell, they can bind to any cell of my body. ठीक है? Ma'am is a really good teacher. Thank you so much, Harshvardhan. Very nice to know that you're liking the lecture. So, Fab portion say the antibody binds an antigen, and Fc portion say the antibody binds the cells of my own body, right? So, what the same thing is happening here. The mast cell has receptors for these IgE's Fc portion. So, now this IgE from its Fc portion, okay, ये तो है, this is the Fab portion which will bind this allergen, okay, and this is the Fc portion from where it will bind to the mast cell, right? Now, what is this mast cell? It is a sensitized mast cell. Now it is sensitized. It has seen, it has tasted these antibodies. It will attach these antibodies and just float about. So this is the first step which is happening, which is mast cell sensitization. So what is the first step of type 1 hypersensitivity reaction? The first step is mast cell sensitization. What is the second step of hypersensitivity reaction? The second step is mast cell degranulation. Mast cell degranulation. This is the second step. ठीक है, यही तो पढ़ा हमने डिफरेंसेस टेबल में. That effector mechanism is mast cell degranulation. So how and when and why will this mast cell degranulate now? You see, this is my mast cell. This is my mast cell. It had all of these granular enzymes inside. And what was the property of this mast cell? That it was sensitized. So this here is a sensitized mast cell. What is the meaning of a sensitized mast cell? That all the receptors on the mast cell have these antibodies, IgE antibodies bound to its surface. That is a sensitized mast cell. It has these IgE bound to its surface, right? Now what will happen? This is a sensitized mast cell. Now a secondary attack will happen. The allergen is a stupid guy. Allergen is a stupid guy. He is not listening. He came back. Okay, so you were allergic to pollen grain. The first time ever a pollen grain entered your body, this is what happened. The pollen grain was presented by APC to your T helper 2. T helper 2 released interleukin 4, spoke to B cell. B cell produced specific IgE. Those IgE went and attached to a mast cell. The mast cell is now sensitized. You have this sensitized mast cell in your body. Again, pollen grain season comes. Okay. See, I have told you that immunological reactions and tests have been covered in the one shot. Microbiology Queen, thank you so much, guys. All right. Yeah. So this sensitized mast cell is in your body. Again, spring season comes. Again, pollen grain comes. Now, this pollen grain will come and it will attach to both of these. ठीक है? Attach to both of these. Right? It will come here and it will attach to both of these. So you know what happens? A head butting happens. Two IgE were sitting. This pollen grain came, took the head of one IgE, took the head of another IgE, bam. Head butting happens. What is that called as? That is called as bringing the two antibodies together. So see how it is binding to both the corners. So it has brought these two IgEs next to each other. As a result, these receptors have come closer. The moment these receptors come closer, the moment the head butting happens, mast cell gets angry, mast cell loses it and mast cell totally degranulates. Now all of these granules which were present inside, they start leaking out. Okay, they start leaking out of the cell. This process is called as what? This is called as degranulation of the mast cell. This is degranulation of the mast cell. As a result, these granules which have come out, they will release prostaglandins, okay, leukotrienes, histamines. So what will prostaglandins, leukotrienes and histamines do? They will lead to allergic response. They will cause bronchoconstriction, they will cause vasodilatation, they will cause increased mucus production, they will lower the BP. Ultimately, these are the mediators of allergic response, are they not? You know it from pathology knowledge that mediators of allergic response are your leukotrienes, histamines and prostaglandins. and now pollen grain has come and you're sneezing uncontrollably, you can't breathe anymore, and your allergic response is over. So what had happened here? This was a secondary exposure. This was a secondary exposure, okay? And what had happened here? This was a primary exposure. Primary exposure. So always write type 1 hypersensitivity reaction in 2 steps. The first step is mast cell sensitization and the second step is mast cell degranulation. Okay. All right, amoeba, I am working on them. 4.0, you will really like them. Chalo. So, mast cell sensitization, then mast cell degranulation. हो गया. Now we have to talk about type 2 hypersensitivity. ठीक है? What happens in type 2 hypersensitivity reaction? Let's talk about it. Type 2 hypersensitivity reaction. What is this? Cytotoxic antibody mediated response. मतलब now antibody dependent cellular cytotoxicity is going to happen. So you have this cell. This is a target cell of my own body. This is a cell of my own body. Okay. It has certain antigens on top of it. This cell has these antigens, ठीक है, कुछ भी इसके ऊपर these antigens are present. So it could be drug allergies, hemolytic reactions. So I am a Rh negative person or I am an Rh positive person and there's a mismatch, a blood transfusion which have happened. You took all the lectures, all right, thank you. Chalo. There is a mismatch blood transfusion and now this antigen, this cell with the antigen has come inside the body. This will be seen by whom? This is antibody mediated, na? Antibody mediated reaction. So here main thing has to be antibody. Antibody बनने के लिए you have to look at B cell activation. So this antigen has come, it has activated the B cell. This is a B cell with the B cell receptor, right? You know what is a B cell receptor? A B cell receptor is itself an IgM or an IgG antibody. You know it, है ना? So the B cell receptor recognizes the antigen on this cell. Now this B cell will activate into a plasma cell. Plasma cell. Plasma cell will lead to antibody production. What are these antibodies which have now been produced? IgM or IgG. What are the antibodies? IgM or IgG antibodies have been produced. Now, how will they cause neutralization or killing? They will cause it by ADCC. What is ADCC? ADCC. Full form क्या है? Antibody dependent cellular cytotoxicity. Antibody dependent cellular cytotoxicity. ठीक है, मतलब the whole process is dependent on an antibody. So now the process is going to start because this IgM or IgG got produced. Ultimately what has to happen? Cell death has to happen. This cell has to die. Cytotoxicity has to happen. Who will cause this cytotoxicity? This cell, cellular cytotoxicity, right? Ultimately a cell is dying. Who is killing the cell? Another cell is killing it. But all of this activation happened because some antibody was produced. Antibody dependent cellular cytotoxicity. So what have we achieved till now? Till now we have achieved antibody production. Antibody बन गई. Now this antibody will go and cover this antigen पूरा. So क्या था? This antigen was there with its ये रहे antigens on top of it. Now these antibodies which are produced will come. They will bind. They will cover it. They will bind to these antigens. All right. Now you know that from the Fab portion, the antibody binds the antigen. But from the FC portion, It will bind to the cells of my body. है ना? This FC portion is what I am interested in. This FC portion will bind to whom? This FC portion will bind to the natural killer cells. This FC portion will bind to macrophages. This FC portion will bind to all the phagocytes of my body. As a result, when this natural killer cell will burst, this whole thing is going to get killed or die. Right? The whole thing dies. So what is happening? Ultimately, this cell is dying. So cytotoxicity is happening. Who is killing this cell? This natural killer cell is killing this cell. So the cytotoxicity is cellular cytotoxicity. It is cell-mediated cytotoxicity. Why did this NK cell start to kill? Because this antibody was produced. This antibody was produced. So this whole killing is dependent on an antibody. So antibody dependent cellular cytotoxicity is the cytotoxic or the type 2 hypersensitivity reaction. Okay? Or there can be complement pathway which can get activated. So the C3b will either cause opsonization or there will be the MAC complex formation. Right? Done. Now type 3 is quite simple. You have seen ADCC. You have already seen complement and opsonization and MAC formation. I am not repeating that. In this lecture itself we have seen. Antigen antibody complex deposition is quite simple. Now finally we have to see the type 4 hypersensitivity. Type 4 hypersensitivity reaction. Okay. So let's talk about type 4. Again we will remember our Anil Kapoor. Mr. Anil Kapoor. What did he say? He said 1 2 ka 4. 1 2 ka 4. Again we will look at that. Here also, in type 4 also, the first step is sensitization. The first step is sensitization. Okay, this is cell-mediated immunity. Here also the T cells are going to get involved. You have an antigen. कौन है ये? This is TB. This is tuberculosis antigen. This is seen by whom? This is seen by some antigen presenting cell. Antigen presenting cell has the bicep MHC. MHC will show this antigen to a T cell, a T helper cell. Which T helper cell now? 1 2 ka 4. What is the hypersensitivity reaction? 4. So now again we have to go back to 1. So what is the T helper involved? T helper 1. What will the T helper 1 release? Interleukin 2. Okay. So what is the T helper here? T helper 1. So obviously the MHC is going to be, the CD is going to be CD8 and MHC is going to be 1. That's okay. Antigen presenting cell shows the antigen to a T helper 1 cell. Okay. Now this T helper 1 cell is activated, it is sensitized. It has all of these receptors which are sensitized for this antigen. What is this T cell now called as? This is a sensitized T cell which is called as delayed type hypersensitivity T cell. Okay, this is an activated sensitized T cell. Now again secondary attack happens. Okay, so now the second step is what? Second step is effector phase. Effector phase means now effect is going to come. When does effector phase start? It starts on secondary attack. So what was this? This was primary attack. First time antigen had come. Now there is secondary attack. Secondary attack. Okay. Example is tuberculosis. For the first time in your life, when Mycobacterium tuberculosis entered your body, it was shown by antigen presenting cell to a T helper 1 cell. The T helper 1 cell had become a sensitized delayed type hypersensitivity cell. Now in my lab, I took a TB antigen and I injected in your forearm. What is that? Montague's test. What is the basis of Montague's test? Delayed type hypersensitivity reaction. Your T cell is already sensitized, it's a delayed type hypersensitivity cell. I have taken TB antigen in an injection injected into your forearm. Now what will happen? This delayed type hypersensitivity was waiting for this secondary attack. This injection of Montux is like a secondary attack inside your body. And now this will release your 1,2-ka-4 Anil Kapoor. This will release your interleukin 2. Interleukin 2. Also it releases tumor necrosis factor. It releases interferon gamma. What do these guys do? They call the killers. This will call cytotoxic T cell. They will call the macrophages and the other phagocyte cells. What will happen? Granuloma formation. How does granuloma formation happen? This cell was really trying to kill it, but it was not able to kill it. It called these macrophages. These macrophages turned into activated macrophages. What are they called as? They are called as epithelioid cells. When many epithelioid cells, they merge together, they form the Langerhans giant cell. All of this is definitely ringing a bell in your mind. What is it leading to? Ultimately, the final step of delayed type hypersensitivity is granuloma formation. Beach mein there is craziating necrosis, surrounded by activated macrophages, surrounded by Langerhans giant cells that has a collar of the T lymphocytes followed by the fibroblasts. Granuloma formation is complete. Skin and soft tissue we are going to do, guys, don't worry. Chalo. So now we are done with all the 4 types of hypersensitivity. We are done with the lecture. So this was all. See, finally, what have we achieved? We have achieved phagocytosis, macrophage. Please remember, contact dermatitis feels like a type 1 hypersensitivity reaction, but it is not. It is a type 4 type of hypersensitivity reaction. Montux test of tuberculosis is an example of type 4 hypersensitivity reaction. That is what we have just seen. All right, guys. So thank you so much. Yes, I will share the notes very soon, and I will see you in the next class. Thank you so much for attending. Bye bye, guys. Take care. Good night.